cellular respiration. Glucose oxidation happens first, which contains glycolysis, link reaction, and the Krebs cycle. All of these are aerobic, meaning they use oxygen. Our first reaction happens in the cytoplasm in a process called glycolysis. In the investment phase, the cell uses ATP to start the process of glucose oxidation. 2 molecules of ATP are broken into ADP and phosphate. This added energy helps split glucose into 2 3-carbon molecules. These 3-carbon molecules are called G3P, also known as pGal. To recap, during this reaction, glucose and 2 ATP go in, and 2 G3P and 2 ADP are produced. Next, we'll enter the energy payoff phase of glycolysis. In the payoff phase, energy is released. Each molecule of G3P is converted into pyruvate or pyruvic acid. As this happens, important things occur. Electron carriers are formed when NAD+ gains electrons, becoming reduced, and then becomes NADH. These NADH molecules will carry electrons to later stages of cellular respiration. Then, 4 ATP are produced by substrate-level phosphorylation. As you can see here, to recap, in goes G3P into NAD+, and 4 ADP. Things that go out are pyruvate, 2 NADH, and 4 ATP. We will then move into the link reaction, also known as pyruvate oxidation. Just know that everything now will happen 2 times. I only show it once, but it happens twice. So pyruvate moves into the mitochondrial matrix. Inside the matrix, electrons are transferred to NAD+, forming another NADH. Here, the 3-carbon fragment attaches to coenzyme A, and 1 carbon is released as carbon dioxide, which forms acetyl-CoA. To recap, in this reaction, in goes pyruvate, coenzyme A, NAD+. Out goes acetyl-CoA, CO2, and NADH. Again, everything that I am about to show happens 2 times. Acetyl-CoA now enters the Krebs cycle, also called the citric acid cycle, which still occurs inside the matrix. Throughout the cycle, acetyl-CoA is gradually broken down. First, acetyl-CoA reacts with a 4-carbon molecule known as oxaloacetate. This makes a 6-carbon molecule called citric acid. Citric acid gets oxidized, transferring electrons and hydrogen to NAD+, forming NADH. This oxidation reaction caused the release of carbon dioxide as well. So now, if you haven't noticed, we have a 5-carbon molecule. This molecule undergoes a similar process of oxidation. This produces another NADH and another CO2. Now, we have a 4-carbon molecule. This 4-carbon molecule undergoes another chemical reaction. The energy released by this reaction is used to join ADP and phosphate to form ATP. This molecule gets oxidized even further, transferring electrons and hydrogen to a different electron carrier called FAD. This forms FADH2. This also carries electrons to the electron transport chain. This 4-carbon molecule gets oxidized one more time, forming even more NADH. The final reaction also results in making our starting molecule oxaloacetate once again. This is why the Krebs cycle is a cycle. We end up right where we started. These reduced electron carriers now carry electrons to the next major stage. A total of 2 ATP are produced in this stage. Oxidative phosphorylation, which includes the electron transport chain and chemiosmosis. The electron transport chain, or ETC, is located along the inner mitochondrial membrane. A vast majority of ATP is made here. NADH, an electron carrier, delivers its electrons to the first protein in the electron transport chain. As the electrons move through this protein complex, energy is gradually released. And this energy is used to pump hydrogen ions or protons across the inner membrane from the matrix into the inner membrane space. The electrons are then transported by the molecule toward their next destination. This molecule also picks up electrons donated by FADH2, the electron carrier that we made during the Krebs cycle. All of these electrons are then passed on to another protein complex. At this next protein complex, the electron energy is used once again to transport hydrogen ions across the membrane into the inner membrane space. By doing this, we build up a high concentration of hydrogen ions in the inner membrane space. Now the electrons are transferred to a final protein complex, where their energy is used once again to pump hydrogen ions into the inner membrane space, making the concentration even higher. Now the electrons need somewhere to go, so they go to the final electron acceptor, oxygen. Oxygen accepts electrons as well as hydrogens from the matrix, making water. We need oxygen for the whole process because it keeps everything running. The proton buildup in the inner membrane space creates a strong electrochemical gradient. Protons flow back into the matrix through an enzyme called ATP synthase. As protons move through ATP synthase, the enzyme rotates and produces ATP from ADP and phosphate. As you can see here, most of the ATP made during cellular respiration comes from this process, as I've said before. This movement of protons is called chemiosmosis. A total of about 28 ATP is produced in this process. Next is fermentation. Fermentation begins when the cell has no oxygen, meaning aerobic respiration can't continue, so we return to glycolysis, which happens in the cytoplasm. Here, glucose is broken down into two pyruvate molecules. A small amount of ATP is produced using substrate-level phosphorylation, and NAD+ is reduced, becoming NADH. But without oxygen acting as a final electron acceptor, NADH cannot drop off its electrons in the electron transport chain. The cell must now recycle NADH back into NAD+, and that is what fermentation does. There would be two pyruvates here, but I'm too lazy to show it, so next we move into alcohol fermentation, which occurs in yeast and some bacteria. Each pyruvate molecule is converted into ethanol, and carbon dioxide is released. As you can see here, during this process, NADH is oxidized back into NAD+, which allows glycolysis to continue producing ATP even though oxygen is unavailable. Again, in this process, there would be two pyruvates. Finally, we transition into lactic acid fermentation, which occurs in animal cells. Here, pyruvate is directly converted into lactic acid. As this happens, NADH is oxidized back into NAD+, once again enabling glycolysis to continue. This process produces no extra ATP. It is purely an emergency pathway used when oxygen is unavailable. Also, no carbon dioxide is produced. Forgot to mention this, but fermentation is anaerobic, meaning it doesn't use oxygen. Now we're going to shift gears a little bit and move into photosynthesis. So we're going to start with light dependent, which consists of electron transport chain and chemiosmosis. So let's zoom in on the thylakoid and focus on the thylakoid membrane. Light-dependent reactions occur along the thylakoid membrane, inside the lumen. This is where pigments such as chlorophyll are found and is where light gets absorbed. The pigments are arranged in clusters called photosystems. Let's start with photosystem 2. When photosystem 2 absorbs light, especially red and blue wavelengths, electrons in chlorophyll gain energy or get excited from the light. These excited electrons leave the chlorophyll and move to something called an electron transport chain. But since chlorophyll lost electrons, those electrons need to be replaced. To replace them, water is split and its electrons go to chlorophyll. When water is split, electrons, hydrogen ions, and oxygen are produced. Now back to the electron transport chain. The electron transport chain transports electrons. As it does, the energy in the electrons is used to pump hydrogen ions or protons across the thylakoid membrane into the thylakoid. This creates a high concentration of hydrogen ions inside the thylakoid. These ions would be trapped inside the thylakoid, but a special enzyme called ATP synthase allows them to passively diffuse from high to low concentration. This flow of hydrogen ions through ATP synthase causes ATP synthase to spin and produce ATP. This ATP is a key product of the light-dependent reactions. Now let's focus back on the photosystems. When electrons reach the end of the first electron transport chain, they go to photosystem 1, where light excites them once again. They move down a second, ETC, where they are accepted by a molecule called NADP plus reductase. When it accepts the electrons, it also accepts hydrogen and becomes NADPH. NADPH is an electron carrier, and it is another key product of the light-dependent reactions. It carries electrons and hydrogens to the next set of reactions in photosynthesis. Both ATP and NADPH are critical products of the light-dependent reactions that are needed to make sugar in the Calvin cycle. Next, we move into the Calvin cycle, also known as light-independent reaction. This contains carbon fixation, reduction, and the regeneration of RUBP. Phase 1 is carbon fixation. In the stroma, the enzyme Rubisco attaches CO2 to RUBP, forming a 6-carbon compound that splits into 2 3-carbon sugars, 3-PGA. This happens for 3 CO2 molecules. The reactants are 3 CO2 and 3 RUBP. The product is 6-3-PGA, aka PG, which I didn't clearly show in my animation, so I just wanted to include that in my voiceover. Next, we move into reduction. So the reactant is 6,3-PGA, which I don't show here, I just show one. ATP provides energy, and NADPH donates electrons, reducing 3-PGA into G3P, the main product of the Calvin cycle. The The product is 6G3P. Again, I only show one. Next, we move to phase 3, regeneration. So 1G3P leaves the cycle, as you can see, becoming glucose. The remaining 5G3P molecules, 15 total carbons, as you can see, 5 times. There would be 5 of these. Again, I only show one. Regenerate 3 RUBP molecules using more ATP. Again, 3 times there would be 15 total carbons, but don't show it. Lastly, I'm going to talk about the connection between photosynthesis and cellular respiration. So photosynthesis stores light energy in glucose. Cellular respiration releases stored energy to make ATP. The oxygen made in photosynthesis is used in respiration, and the carbon dioxide from respiration is used in the Calvin cycle. Together, these processes cycle matter and energy through living systems.